Usually when you picture the origins of modern medicine, you imagine something pretty pristine.
Oh, for sure. Gleaming white labs, hazmat suits.
Right. Sterile environments, pharmaceutical breakthroughs, engineered down to the atom. We want to think of medicine as this highly controlled thing, completely divorced from the messy reality of the natural world.
It's a very comforting image, honestly. We want life-saving treatments to feel deliberate, technologically advanced from the very start.
But when you look at the actual source material for some of our most critical drugs, that pristine image just vanishes, and it gets replaced by literal mud.
Yep, dirt.
So welcome to the Deep Dive. Today, we are exploring a stack of research on a class of drugs called tetracyclines.
And this isn't just some dusty historical pharmacology footnote.
Not at all. It is a load-bearing pillar of modern medicine. It's a drug class that connects to bioterrorism preparedness, climate change, and some incredibly bizarre physical side effects.
The side effects are wild.
Yeah, we're going to get into that. Our mission today is to understand how these broad-spectrum antibiotics mechanically dismantle an infection, the very specific reason they can permanently stain human teeth, and the history of how we even found them.
Which is a crazy story.
It really is, because the history starts with this fascinating paradox from our sources. We have to figure out how a 71-year-old retiree just digging through dirt in Missouri ended up discovering the foundation for a drug that currently sits in our strategic national stockpile for mass anthrax exposure.
The contrast there between the origin and the modern application, it's just staggering. But to fully grasp what this drug does inside the human body today, we really have to understand the historical pressure that sent researchers digging into the ground in the first place.
Okay, let's set the scene. The year's 1945. World War II has just ended. Penicillin has been this massive wartime miracle, but it has severe limitations.
Right. Penicillin was groundbreaking, but it had a very narrow spectrum of activity, mostly working against gram-positive bacteria.
Which means what, exactly, for the listener?
Well, imagine bacteria as tiny fortresses. Gram-positive bacteria have a thick outer wall, like a heavy winter coat. Penicillin is incredibly good at destroying that specific coat.
Okay, but there are other types.
Exactly. You have gram-negative bacteria. They have a much thinner wall, but they surround it with an extra outer force field, a lipid membrane. And penicillin just bounces right off that force field.
Oh, wow.
Yeah. And beyond those two, you have atypical organisms that just don't conform to either structure.
So doctors were essentially watching patients die of infections that penicillin couldn't even touch. I mean, they had streptomycin, which was discovered a couple of years earlier, but the sources note it was highly toxic.
Highly toxic, and it had to be injected.
Right. So the medical world was desperate for an oral drug, something you could just swallow that would act like a shotgun instead of a sniper rifle. They needed a broad-spectrum weapon.
And researchers realized that soil bacteria had already been fighting these microscopic chemical turf wars for millions of years.
Just constantly attacking each other in the dirt.
Exactly. They were constantly secreting chemical weapons to kill their neighbors and steal resources. So humans didn't actually need to invent a broad-spectrum weapon from scratch. We just needed to find the organism that had already evolved one.
Which brings us to Benjamin Minge-Duggar. He's a botanist, a plant pathologist. And at 71 years old, a time when most people are, just enjoying retirement, he's working at Ludural Laboratories.
Meticulously screening thousands upon thousands of soil samples from across the country.
Yeah. And in a specific sample of Missouri dirt, he isolates this unique, golden-colored microorganism. He names it Streptomyces aureofaciens, which translates to gold-making.
A highly prophetic name, honestly, because by 1948, Duggar managed to isolate a specific compound from that golden microbe. It was brought to market as chlortetracycline or aureomycin.
The Holy Grail.
Literally, it was the first true broad-spectrum antibiotic. Suddenly, a doctor could hand a patient a single pill that covered the gram-positive fortresses, the gram-negative force fields, and even those strange atypical organisms.
And that discovery just kicks off a frantic corporate gold rush. The pharmaceutical industry realizes the dirt is full of billions of dollars. So companies like Pfizer scrambled to compete. They're literally paying missionaries, soldiers, pilots to just mail them soil samples from all over the globe.
Yeah, mail us your dirt.
It's crazy. And by 1950, Pfizer manages to isolate a slightly different compound, oxytetracycline, from a completely different soil microbe. But then a Pfizer chemist named Lloyd Conover does something that fundamentally changes pharmacology.
Right. He doesn't just purify what the mold naturally produces.
No, he looks at this chemical structure and decides to aggressively remodel it.
He took the natural molecule and subjected it to a process called catalytic hydrogenolysis.
Okay, let's break that term down for a second for the listener, because the mechanism is pretty brilliant.
Think of it as using hydrogen gas and a metal catalyst to act like molecular scissors. Conover used this process to snip a single specific chlorine atom off the chlorotetracycline molecule.
So he strips away a piece of nature's design.
Exactly, leaving the core structural weapon completely intact, but making the drug more stable. He engineers tetracycline itself.
That is a monumental shift. We went from simply finding and purifying natural products to taking nature's blueprints and actively re-engineering them in a lab.
It really was the birth of semi-synthetic antibiotics. Conover's molecular scissor work was so revolutionary that it eventually earned him a spot in the National Inventors Hall of Fame.
Wow.
Yeah, he proved that human intervention could improve upon millions of years of microbial evolution.
So Conover proves we can chemically engineer these natural weapons. But how does that weapon actually fire? When you swallow this drug, how does it stop a massive, life-threatening infection?
Well, the sources are very specific in pointing out that calling tetracycline a broad-spectrum killer is entirely inaccurate.
Right, because it doesn't actually kill the bacteria.
No. And that distinction brings us to the concept of selective toxicity. The holy grail of any drug is finding a way to destroy the invader without harming the human host.
Makes sense.
To do that, you have to target a piece of machinery that the bacteria possess, but human cells do not. And tetracyclines target the ribosome.
The ribosome being the cellular factory that builds all the proteins an organism needs to survive.
Precisely. Bacterial ribosomes are structurally distinct from human ones. Bacteria use what are called 70S ribosomes, while human cells use larger 80S ribosomes.
Okay, so because of that structural difference in the factories, the drug can safely flood human tissue, totally ignore our cells, and zero in exclusively on the bacterial machinery.
Exactly. And the key to how it infiltrates that specific bacterial factory is in its physical shape. The name itself, tetracycline, gives away its structural signature. It consists of a 4-ring carbon skeleton.
A naphthecene core.
Yes, specifically called a naphthecene core. That 4-ring skeleton is the physical wedge. The bacterial 70S ribosome has a lower half called the 30S subunit. On that subunit, there's a very specific docking bay known as the A site. In a healthy, functioning bacterium, molecules called aminoacyl tRNAs arrive at this A site, carrying fresh amino acids, which are the raw materials needed to build a new protein.
If we visualize this as a factory assembly line, the messenger RNA is the conveyor belt sliding along, giving the instructions.
Right.
And the tRNA molecules are the factory workers rushing up to the loading dock, the A site, carrying the new parts to build the product. So what that 4-ring tetracycline molecule does is it essentially parks a massive truck right inside the loading dock.
That is the exact mechanism. The truck is parked in the A site. The workers arrive with the raw materials, but there is nowhere to unload them. Protein synthesis completely halts.
The factory doesn't blow up. The bacterium doesn't rupture or explode. The assembly line just completely stops moving.
Which is why the term killer is a misconception. Tetracyclines are bacteriostatic.
Meaning they stall bacterial growth and reproduction.
Exactly, rather than being bactericidal, which would mean they outright kill the cell. By parking that truck in the loading dock, the bacterium can no longer replenish its vital enzymes, it can't repair its cell wall, and it cannot divide to multiply.
It's essentially freezing the enemy in place. But that implies the drug alone isn't enough to cure you. You need a functioning immune system to come in and sweep up all those stagnant frozen bacteria.
That is a critical clinical reality. It is exactly why physicians generally avoid prescribing bacteriostatic drugs to severely immunocompromised patients.
Because if their white blood cells can't show up.
Right. If they can't clear away the frozen bacteria, the infection will just resume the moment the drug leaves the system. Though, pharmacologists will often add a layer of nuance here. The strict division between static and cytal drugs is sometimes just an artifact of how we test them in lab cultures.
Meaning, what happens in a glass test tube isn't always exactly what happens in a human body.
Yes. In a test tube, the drug concentration is fixed, and we observe that it just stalls the bacteria. But in human tissue, drug concentrations rise and peak after you take a pill. And depending on the specific strain of bacteria and the dosage, that stalled factory can occasionally undergo enough stress to cause cell death.
Interesting.
But fundamentally by design, it is a jammer, not an explosive.
The precision of that 4-ring shape how it perfectly wedges into the loading dock is brilliant. But according to the sources, that exact same structural shape has a fatal chemical flaw.
It does.
It possesses an irresistible chemical attraction to calcium.
The scientific term for that attraction is chelation. Tetracyclines bind to heavy metal ions, particularly calcium ions, with an incredibly high affinity.
And this seemingly minor chemical quirk led to a massive visible medical discovery in the 1950s and 60s. Pediatricians and dentists started noticing a highly alarming pattern in their clinics.
Oh, yeah.
Young children who had been treated with early tetracyclines for infections were developing permanent yellow, gray, and brown bands across their teeth.
Because of that powerful chelation. When the drug encounters calcium in the body, it locks onto it. It forms a tightly bound tetracycline calcium orthophosphate complex.
And during childhood, as secondary adult teeth are growing and mineralizing under the gums, they are pulling in large amounts of calcium.
So the body accidentally pulls the drug calcium complex right into the tooth.
It's not just a surface stain like you'd get from, say, drinking too much coffee.
No, it is a fundamental architectural change. The complex gets physically integrated right into the hydroxyapatite crystal lattice of the developing enamel and dentin. It literally becomes a structural building block of the child's tooth.
The visual element of this is striking because that integrated chemical complex reacts to ultraviolet light.
Yes.
When a child with these built-in stains smiles in the sun, the UV exposure actually causes the chemical bands to oxidize and darken over time.
It's terrible.
And because this drug blindly hunts for calcium wherever it's being rapidly deposited, it doesn't just stop at teeth. It aggressively binds to actively growing bone tissue, which temporarily inhibits bone growth in premature infants.
And this physiological reality fundamentally dictates modern prescribing guidelines. It remains a strict contraindication for pregnant women because the drug easily crosses the placenta and will bind to the developing fetus's bones.
Right.
And it is strictly avoided in children under the age of 8 whose secondary adult teeth are still actively calcifying.
But once you're an adult,
Once you're an adult and your teeth are fully formed and mineralized, the risk of structural integration is gone. But during development, the damage is permanent.
if the core chemical skeleton of this drug has a fatal aggressive attraction to calcium, shouldn't we be extremely worried about taking it with a glass of milk? Like if you swallow a tetracycline pill and wash it down with dairy, or if you just took a calcium-based antacid for heartburn, doesn't the drug just bind to the calcium in your stomach and become totally useless?
You've just hit on one of the most enduring misconceptions in modern medicine.
Around.
For the older, original generations of the drug, like the ones Duggar and Conover worked with, you are entirely correct. The calcium in dairy or antacids would instantly chelate the drug in your gut, forming a heavy complex that your intestines couldn't absorb, effectively ruining the treatment.
But that's not true anymore.
Not for the versions we primarily use today. Modern medicine largely relies on newer generations, specifically a drug called doxycycline.
Okay.
The molecular structure of doxycycline was tweaked to be highly lipophilic, meaning it dissolves readily in fats and is absorbed so efficiently by the digestive tract that a normal amount of dairy or food hardly impacts its clinical effectiveness.
Wow.
Yet, if you ask almost anyone, they will tell you to never take any tetracycline with milk. It is a fascinating case where clinical habit and old advice have vastly outpaced actual pharmacokinetic data.
That is wild. The advice outlives the science entirely. So if pregnant women and young children are excluded from taking this class of drugs, we need to look at who's actually taking it today, because the sources show that millions of adults rely on these drugs for a surprisingly diverse, almost bizarre set of reasons.
Oh, yeah, really do.
Examining these different use cases exposes the quirky personalities of the different generations of the drug.
Absolutely. Doxycycline is undoubtedly the modern MVP of the entire class. Because it retains that broad spectrum ability, it is highly effective against atypical bacteria, parasites, and a specific spiral-shaped type of bacteria called spirochetes.
Which brings us to a massive public health application: Lyme disease.
Exactly. Lyme disease is caused by a spirochete transmitted through tick bites. If you've ever been treated for Lyme, you almost certainly took a course of doxycycline.
There is a landmark study from the sources that highlights just how powerful this drug is in that specific scenario. It's the Nadelman study, published in the New England Journal of Medicine in 2001.
A classic.
Right. Researchers wanted to know if doxycycline could be used as a prophylactic, essentially stopping the disease before it even fully establishes itself in the body. They found that if a patient was given just a single 200 milligram pill of doxycycline within 72 hours of removing an engorged deer tick, The rate of developing the classic Lyme disease rash plummeted from 3.2% in the placebo group down to just 0.4%.
One single dose of a drug blocking the spirochetes before they can disseminate through the tissue.
It's an astonishing risk reduction.
It is. And it is the exact reason why a single dose of doxycycline is the standard post-tick bite protocol in emergency rooms today.
It's treated like a miracle drug for outdoorsmen. It goes further than that. It's the first-line treatment for Rocky Mountain spotted fever. It clears severe acne. Travelers take it daily as malaria prophylaxis. And, circling back to our introduction, the government stockpiles literal tons of it.
Yep.
In the event of a mass bioterrorism attack involving weaponized anthrax or plague, doxycycline is the drug they will be handing out to the population. It really acts like the Swiss Army knife of antibiotics.
It is remarkably versatile. But as you mentioned, the other generations in this drug class have some very strange personalities. Take the minocycline.
Okay.
It is rarely prescribed for bacterial infections anymore. Instead, physicians exploit one of its strange side effects. The minocycline induces a condition called diabetes insipidus.
Mechanistically, how does an antibiotic do that?
It physically blocks the receptors in your kidneys that respond to antidiuretic hormone, or ADH.
And ADH does what?
ADH is the signal your brain sends to tell your kidneys to hold on to water. Demeclocycline blocks that signal, essentially forcing the kidneys to ignore the brain and continuously dump water into the urine.
So doctors intentionally use this drug to make people lose water.
Yes. They use it off-label to treat a completely separate condition called SIADH, where a patient's body incorrectly hoards dangerously high amounts of water, which dangerously dilutes their blood sodium levels. By administering an antibiotic that blocks the water retention receptors, they force the kidneys to pee out the excess fluid. They are using the drug's side effect as a highly targeted cure.
That is brilliant pharmacological judo.
Right.
And then the sources detail minocycline, which has its own extreme quirk. You mentioned earlier that modern versions are lipophilic, meaning they dissolve easily in fats. Minocycline is highly lipophilic.
Very much so, which allows it to easily cross biological barriers that protect the central nervous system. It can cross into the vestibular system in the inner ear, which frequently causes severe vertigo and dizziness in patients.
And minocycline is the one with the extreme pigmentation side effects, right?
Yes. Because it is highly lipophilic, it penetrates deep into tissues. With long-term use, which is very common when dermatologists prescribe it for severe acne, minocycline can actually oxidize and turn a patient's skin, their gums, the whites of their eyes, and even old physical scars a distinct bruised blue-gray color.
That is unnerving.
And beyond the cosmetic, there is a rare but serious mechanical side effect associated with long-term use, mostly seen in young women treating acne. It is linked to idiopathic intracranial hypertension.
Intracranial hypertension, meaning a dangerous buildup of pressure inside the skull.
Precisely. The drug can trigger an overproduction or underabsorption of cerebrospinal fluid. The fluid builds up, putting immense physical pressure on the brain and the optic nerves, causing severe headaches and potential vision loss.
Speaking of physical damage caused by the pill, the sources highlight a highly practical, terrifying danger regarding how you actually swallow the medication.
Oh, this is important.
If you take a doxycycline capsule right before laying down for bed, and you don't drink a large glass of water, the pill can get physically lodged against the wall of your esophagus before it reaches the stomach.
A very common, very painful mistake.
The chemical acidity of the drug sitting directly against the tissue will literally burn a hole, an esophageal ulcer, right into the throat. You essentially chemically burn your own esophagus because you didn't drink enough water.
Just drink the water.
Right. Which leads into another logistical issue with these pills: expiration dates. We all have that old, dusty bottle of antibiotics sitting in the back of the medicine cabinet that expired 3 years ago.
Oh, definitely.
But ignoring the expiration date on a tetracycline caused a massive systemic scare in the 1960s.
The Fanconi syndrome scare. If there is ever an expert cautionary tale about why expiration dates are more than just a manufacturer's liability suggestion, this is it. In the early 1960s, physicians began seeing terrifying clusters of patients developing Fanconi syndrome.
What exactly fails in the body during Fanconi syndrome?
It is a profound failure of the proximal tubules in the kidneys. These tubules act like the body's recycling center. Their job is to filter the blood and pull essential nutrients like glucose, amino acids, and bicarbonate back into the bloodstream so you don't lose them.
Okay.
In Fanconi syndrome, the recycling center breaks down, and all of those vital nutrients just leak out of your blood and pour straight into your urine.
And they traced this catastrophic kidney failure back to old medicine cabinets.
They did. They found that patients were taking expired tetracycline capsules. As the drug ages past its expiration date, the complex molecule naturally degrades. Those degraded breakdown products become highly toxic to human tissue. Yeah, they travel through the blood, arrive at the kidneys, and physically attack the cells of the renal proximal tubule, completely shredding the recycling center.
So the breakdown of the chemical weapon turns it against the human host.
Mm,
It is a stark reminder not to mess around with expired prescriptions.
absolutely.
So we have this incredible versatile toolkit. We have the modern MVP in doxycycline. We have the quirky, highly specialized siblings. But the sources point out that human behavior has severely jeopardized the future of this entire class of drugs.
Unfortunately, yes. We have entered the resistance era. And to truly understand how we compromise this miracle drug, you have to stop looking at hospitals and start looking at farms.
Farmed.
For roughly 70 years, vast quantities of tetracyclines, especially the older generations like chlortetracycline, were mixed directly into livestock feed at subtherapeutic doses.
Meaning they weren't giving the animals enough of the drug to cure an infection. They were just giving them a little bit constantly.
Purely for agricultural growth promotion. Giving livestock a low, constant dose of antibiotics subtly alters their gut microbiome in a way that makes the animals grow larger and faster on less food. It was purely an economic decision.
70 years of just dumping a foundational medical pillar into the food supply to make chickens and pigs slightly bigger.
Yep.
And bacteria, being the ultimate survivors, adapted. The sources explain that this agricultural overuse aggressively drove the spread of TET-resistance genes.
Exactly.
These are mobile genetic elements. Bacteria don't even have to be the same species to share them. They essentially hand blueprints to each other like microscopic flash drives, teaching each other how to defeat the drug.
And those genetic flash drives carry the blueprints for two primary mechanical defenses. The first is the efflux pump.
How does that work?
The bacteria build cellular bouncers in their outer membrane. The moment a tetracycline molecule enters the cell, the efflux pump grabs it and physically ejects it back outside before it can ever reach the ribosome.
The truck never even gets close to the loading dock.
Right. And the second defense relies on ribosomal protection proteins. The bacteria build specialized proteins that travel to the ribosome and physically alter the shape of the A site.
Wow.
They change the lock on the loading dock, so the four-ring structural wedge of the drug just no longer fits.
And because these resistance blueprints are highly mobile, they spread from agricultural runoff into the soil, into the water, and straight into the human pathogens that end up in our emergency rooms.
It's a huge problem.
But the pharmaceutical chemists didn't just surrender. In 2005, they approved a new drug called tigecycline. It's part of a new subclass called glycylcyclines. They essentially took the classic tetracycline skeleton and bolted this massive, bulky chemical side chain onto the 9th carbon position.
It was an act of pure chemical engineering designed to directly counter the bacteria's new defenses. That new side chain makes the molecule physically too large and bulky for the bacterial efflux pumps to grab onto and spit out.
Nice.
And the modified structure allows it to bypass the ribosomal protection proteins and wedge into the A site anyway.
They outsmarted the resistance. I mean, it sounds like a total triumph of human ingenuity. But looking at the clinical data in the sources, did this chemical fix actually save the day for the patients taking it?
That is the crucial question, and the answer is a very sobering reality check for modern medicine. In a test tube, tigecycline was an absolute masterpiece. It defeated the most resistant bacteria we had. But in 2010, the FDA pooled all the clinical trial data and was forced to issue a severe black box warning.
A black box warning is the strictest alert the FDA can put on a drug. What happened?
They discovered that patients receiving tigecycline actually had a significantly higher all-cause mortality rate than patients receiving older standard antibiotics, particularly in cases of severe pneumonia.
Wait, the patients who were given the brand new engineered superdrug died more often.
Yes. It is a harsh, humbling lesson in the limits of pharmacology. Expanding a drug's ability to evade resistance in the controlled, isolated environment of a lab does not guarantee that the chemical will have a favorable risk and efficacy profile inside a complex, living, breathing human being. A chemically upgraded antibiotic is not automatically a clinically better one.
That is incredible. So let's synthesize this entire journey for everyone listening. We started with a 71-year-old retired botanist methodically scooping up thousands of dirt samples in Missouri. From that mud, human ingenuity crafted a semi-synthetic marvel.
A true marvel.
It is a drug that mechanically jams the assembly lines of bacterial factories without blowing them up. It's a chemical with such an affinity for calcium that it irreversibly builds itself into the architecture of our children's bones and teeth. It cures Lyme disease with a single pill, induces water loss by blocking kidney receptors, turns human tissue blue-gray, and stands as a foundational defense in our national bioterrorism stockpile.
And the reason understanding this drug matters to you right now is that its story is actively evolving.
Yeah.
As climate change steadily warms the environment, it is actively expanding the geographic ranges of ticks. Lyme disease is aggressively moving into new territories and states that have historically never seen it. That means doxycycline is becoming more vital to everyday public health and primary care than it has been in decades.
Which leaves us with one final thought to consider. We just spent this entire time dissecting the incredible medical legacy that originated from a single sample of Missouri dirt isolated by one man looking for a weapon in a microscopic turf war.
Right.
If these microbes have spent millions of years evolving complex chemical weapons in the soil, what undiscovered antibiotics are currently waiting for us in the extreme environments we haven't comprehensively checked yet?
It's a fascinating question.
Could the next major class of life-saving drugs, the ones that will bypass our current resistance crisis, be sitting right now in the permafrost, or at the bottom of a deep-sea hydrothermal vent, or deep within the Amazon? It really makes you wonder what kind of miracles are just waiting in the mud if we only take the time to look for them.